we were interested because there were a number of studies showing the relevance of brown adipose tissue also for human metabolism, how this is regulated under conditions of sustained cold exposure. And what was known when we started this experiment is summarized here in this model. So cold exposure, of course, activates the sympathetic nervous system. And at the terminal nerve ends of the sympathetic nervous system, norepinephrine is released. And this activates adrenergic receptors, which then leads to a signaling cascades which activate lipases within the brown adipose tissue. And these leads to the release of free fatty acids from the existing lipid droplets. And these are then transported into the mitochondria for beta oxidation on the one hand. And on the other hand, these free fatty acids also activate the unique protein UCP1, which then generates heat by uncoupling the proton gradient at the inner mitochondrial membrane from ATP production. So, of course, as every heating system, a lot of energy is used for this process, and that is why you need a lot of glucose uptake, fatty acid uptake, and these fatty acids are mainly delivered by triglyceride-rich lipoproteins, because it has been described that lipoprotein lipase is expressed by these brown adipocytes as in white adipose tissue. But we were interested about the exact mechanisms. And first of all, we started an experiment where we used ApoA5 knockout mice just as a model of hyperlipidemia. And in our first experiments, we just simply exposed these mice to cold. And here you see these mice exposed to warm conditions. And when you just simply expose them to cold, you see the clearance of these triglyceride-rich lipoproteins. And here you see triglyceride levels about 2000 milligram per deciliter, which are completely normalized just by simple cold exposure. And we did, after we saw this, we saw, okay, this we have to study in more detail, and we did a number of studies. And here I just want to show you, because meanwhile, a lot of people are looking also for alternative energy substrate for brown adipose tissue. And we did a number of studies with tracers. And what you see is that triglyceride by far is the most important energy substrate for brown adipose tissue. Here you see by an OGFT that glucose, of course, is also taken up in the cold, but still the triglycerides are much more important. And if you analyze it by tracer analysis, you see also free fatty acids are internalized by, especially under cold conditions, the triglycerides are the most important substrate for brown adipose tissue activity. Of course, this is always the question, because the amount of brown adipose tissue in humans is quite limited in comparison to rodents, whether brown adipose tissue also play a relevance in humans. And here I just want to, this is not our work, I briefly want to show you a very recent article from Nature Medicine showing that brown adipose tissue is associated with cardiometabolic health. So from the Paul Cohen group, there was a study where they analyzed more than 50,000 participants in the study, and they did a propensity score matching, and then analyzed people with brown adipose tissue and compared them to matched controls without brown adipose tissue. And then they, of course, they checked for the association of brown fat with cardiometabolic health. And what they did find that there was a huge differences with regard to the type 2 diabetes, dyslipidemia, but also cardiovascular disease. And you can see here the p-values that the presence of brown fat has a beneficial effect with regard to cardiometabolic disease. And if we also look for the energy substrates, we also do see that people with brown fat, that they do have lower glucose level, but also, and this has not been shown before in this so clear, also for triglycerides, indicating again that triglycerides might be also here the trigger for the development of cardiovascular disease, because the number of LDL particles is very similar. So it's mainly due to the effects on glucose and triglyceride levels. Okay, these studies can be summarized in this model. So our studies, we did a number of studies where we analyzed this in much detail, where we also showed that lipoprotein, lipase, and CD36 is essential for this process. We also show that this influences HDL metabolism, so that we have a flux of cholesterol under cold exposure conditions to the liver. We also saw effects on cholesterol conversion to bile acids by the alternative signaling pathway. And this also influences the gut microbiome, which in turn then again leads to the activation via the bile acids to the brown adipose tissue. So there seems to be a very systemic control of this pathway. However, when we saw these kind of pathway, we always were struggled by the fact that brown adipose tissue activation is an catabolic kind of conditions, whereas a postprandial condition is an anabolic condition. And we always wondered, how is this regulated in the brown adipose tissue, and which hormones are under these conditions are important for the uptake. And based on a study which was performed in humans, and this was the only picture, the only figure of this publication by the Cypress group in Cell Metabolism, where they compared the activation of brown adipose tissue between cold and mirabegron. And mirabegron is a drug which is an agonist for beta-3 adrenergic receptors and is used for overactive bladder syndrome. And that is an approved drug. And you can see that also under this condition, using this PET-CT analysis, that there's much higher uptake of glucose into this brown adipose tissue and even higher than in cold. And we were specifically fascinated by the supplemental data, because what you saw is that under these conditions, they do have higher free fatty acid levels, which can be explained by lipolysis in white adipose tissue, but they also do have higher insulin levels and C-peptide levels, indicating that under these catabolic conditions, you have increased insulin levels. And based on this data, we developed a model where we thought, okay, cold exposure might lead, of course, to lipolysis in the white adipose tissue. And these free fatty acids can maybe, because this has been described before, lead to the increase of insulin secretion by pancreatic beta cells, which then in turn could mediate an anabolic response even under cold exposure conditions in the brown adipose tissue. And of course, in addition to free fatty acids, they should be also under these conditions be transported to the liver, because huge amounts of free fatty acids are not transported directly to muscle or brown adipose tissue or the heart. They are transported primarily to the liver and then are used for the generation of triglyceride-rich VLDL in this part. And of course, depending on whether we are on the fasted or on the postprandial condition, this then would lead to the production of these triglyceride-rich lipoprotein, which are then the substrate of CD36 and LPL, which are activated by insulin. And this we then checked, of course, also by experiments. And here you can see the time after CL treatment. And here, just to give you an impression, you can see the liver triglycerides. So there's a stepwise increase in the liver triglyceride in response to CL treatment, indicating that the free fatty acids from the white adipose tissue are delivered to the liver. And interestingly, you see a huge increase in the insulin levels already at 30 minutes. So, of course, this somehow indicates that this is working, and that is why we then thought about how to do intervention studies. And first, we stopped ATGL activity, because then in the cold and or by CL treatment, this should block this pathway. And this actually are the results, what we saw. After CL treatment, you do not see in the in the knockout mice for ATGL, which is specifically in the adipose tissue, that there's no free fatty acid release anymore. And this is a control condition where you can find free fatty acid in the plasma compartment, but this was blunted in the Cree plasmids. And this was associated with a lack of lipid accumulation in the liver. And also, we do not see any insulin increase anymore, indicating that the lipolysis reaction in the white adipose tissue is mandatory for the insulin secretion under these conditions. And then, of course, we looked also whether this somehow influenced brown adipose tissue uptake of energy substrates. And we used different models. And here I will show only the alloxan models where you can deplete the beta cells in the pancreas. And here you see the results. So after alloxan treatment, of course, you have kind of a lack of insulin. That is why your basal levels of free fatty acids are higher. And this is even more pronounced after CL. And of course, you would expect that you have much lower insulin levels. And most importantly, the uptake of lipoprotein particles into the brown adipose tissue, which you can see here under control conditions after CL treatment, the massive increase is totally blocked after alloxan treatment, indicating that you need an insulin response for the uptake of these postprandial lipoprotein particles. And then we did some more studies where we block the insulin receptor signaling. I will not show you all of these data, but just to give you an impression, here we use an insulin and a receptor antagonist, peptide known as S961. And if you treat these mice with these receptor antagonists, then you can see that glucose uptake under this CL condition is really diminished. And the same is true also for the triglycerides. And what is also interesting is that after this kind of treatment, you have also less energy expenditure, showing that energy uptake either by glucose or fatty acids is very important to maintain high energy expenditure rates after activation with the CL compound. Okay, so based on this data, we then propose this model that we have under cold conditions or CL treatment, you have lipolysis, secretion of insulin, which activate, and this we also show by translocation, the CD36 and also LPL activity, and this then leads to higher uptake into the brown adipose tissue, especially of lipids. In the second part of this presentation, I now want to briefly come to the title of my talk, because of course you know that not only in the white adipose tissue or in the muscle, but also in the brown adipose tissue, you have a closed endothelium. And we always wonder how these energy substrates can enter the brown adipose tissue if you have this closed endothelium, and what is the role of the endothelial cells for lipid and also glucose uptake. And to visualize these processes, we have established methods in my lab where we use nanoparticles. Superparamagnetic ones, which are, as you can see here by this picture, magnetic, but also quantum dots, which are fluorescent. And these are so fluorescent that you can do even in vivo imaging. And the other good thing about nanoparticles, at least for our work, is that they are highly hydrophobic. That means you can embed them into the core of a lipoprotein. And this we did here. Here you can see our recombinant lipoproteins at visible light. So they are milky like the plasma I have shown you. And if you then UV excite these recombinant lipoproteins, then you can see that they are highly fluorescent. And this we use for a lot of different studies. But here I just want to show you a picture how these lipoproteins are internalized into activated brown adipose tissue. And what you can really appreciate or easily appreciate from these pictures is that these nanoparticle labeled lipoproteins are internalized predominantly by endothelial cells. And this is a marker for endothelial cells, GPI-HBP1. And here you can see the co-location of our lipoprotein with these particles. And here you do see the multilocular brown adipose tissue. So actually, every brown adipocyte has also contact to these particles. Of course, we do not know whether they are just associated or whether they are internalized by this resolution. And that is why we did some electron microscopy. And here you see the other beauty of these nanoparticles. They have a very high contrast, which you can also visualize by electron microscopy. So here, again, the model, these are the lipoproteins, and we can embed these nanoparticles into the core of the lipoproteins. And then we follow the fate of these particles. And here you can see the brown adipocytes, and here are the endothelial cells. And here you obviously see our lipoproteins containing these nanoparticles. And after a while, you can see that they attach. And if you do a higher magnification, you see this kind of invaginations and also the internalization of after they seem to get smaller during the processing, but then you can see the entire particles also in endocytotic compartments within the endothelial cells of the brown adipose tissue. So when we submitted this paper, then we got a lot of criticism because this is an artifact and you don't show really endothelial uptake. This can't be. Lipoprotein particles are only internalized by the liver. But of course, this somehow indicate for us, and we believe that really lipoprotein particles were internalized because these nanoparticles, they cannot be released by lipoprotein lipase because they are inert. And that is why we did a lot of studies using these kind of particles. And one study is shown here, where we use the magnetic properties. So we isolate the brown adipose tissue after the injection of these particles, which we injected into warm or cold exposed mice. And then we harvest the organs, make a tissue lysate, and did a magnetic pull down and did some stainings via Western blotting. And this you can see here. So here's the input and here's the pull down. And you can see that only if we use our magnetic lipoproteins that we can pull down endosomal and lysosomal compartments, which are positive for RAP5, for instance, RAP5A positive endosomal compartments or LAMP2 positive lysosomal compartments, whereas other compartments remain negative, indicating that really the entire lipoprotein particles are internalized into the endolysosomal system. And this we followed up then in more detail. And in this case, we do not use our recombinant lipoproteins because we always get also the criticism that recombinant lipoproteins are maybe not really mimicking real chylomicrons. And that is why we did a very complicated method to isolate native chylomicrons from rat, which are labeled with radioactive tracers for retinol as well for the triglycerides. These particles are then injected into warm and cold exposed mice. And then we did a max-based cell sorting on the different cell types. So we isolate macrophages, endothelial cells, and the brown adipocytes. And if you then analyze the amount of radioactivity within the different cell types, you can see, so the blue one is always the cold exposed mice and the gray bars are the controls. You can see that there's a very high uptake of our lipoprotein particles, especially in the endothelial cells, while because the triglycerides are processed also to free fatty acids, you can then see that the free fatty acids, so the TG-derived free fatty acids, are primarily then present in the brown adipocytes. So it seems, based on these results, that our lipoprotein particles are hydrolyzed by lipoprotein lipase, that these free fatty acids that are released in the vascular endothelium are internalized by CD36 and are somehow transported via the endothelial cells to the brown adipocytes as energy substrates, while we also observe high amounts of lipoprotein particles in the endothelial cells. And of course, this is now the question, what is the relevance of this process for brown adipose tissue functionality. And you may know that the rate limiting step for the processing within lysosomes for triglyceride and also for cholesterol esters is an enzyme, another lipase, which is known as lysosomal acid lipase. And that is why we then did is generate mice which lack lysosomal acid lipase specifically in the endothelial cells. And with these mice, we then did a number of studies to analyze BAT remodeling and activity and also BAT browning. And this is shown here. And we set up a system where under short-term cold exposure, we check for BAT remodeling and activity, and for a long sustained cold exposure for BAT browning and activity. And this here you see the results of the acute and chronic cold, and you can appreciate that the mice lacking lysosomal acid lipase in endothelial cells, they had less energy expenditure, and they have also lower uptake of glucose as well as triglycerides. And the same is true in white adipose tissue under chronic cold conditions, which you can see here. And also the energy expenditure is somewhat lower in the mice lacking and lysosomal acid lipase in endothelial cells. And then, of course, we were intrigued by this data and checked for some changes which could explain these differences. And what we saw is that it seems that lipoprotein processing within endothelial cells is necessary for efficient browning during chronic cold conditions. And here you can see histology. So you see that here is much more white adipose tissue left compared to the control mice, where you see this browning. And this was also mimicked by gene expression patterns, where you see much less UCP1 and also mitochondrial genes and specific markers, just as deiodinase 2 and alophyl 3, which are lower in the knockout mice. And these are markers for brown and beige adipocytes. This we can also visualize that we thought, okay, this might then explain that you have a lower vascularization, so that we thought that this lipoprotein particle uptake is important for endothelial growth. And this you can see and appreciate by the staining here, where you have much less CD31 positive cells after acute cold exposure in brown adipose tissue and prolonged cold exposure in the white adipose tissue compared to the controls. Of course, we then were asked, what is the mechanisms? And of course, you probably know that endothelial proliferation is highly depending on HIF1 alpha, which is a master regulator for endothelial proliferation under condition of hypoxia. And this is because you have higher expression of a number of genes. I will not go into the detail because this was presented by the Nobel Prize winners. But of course, we were fascinated by this fact that this may be an explanation why we do see some difference. And there was only one description that lipids really may be important for the activity of HIF1. And this was a high-fat diet study, the short-term high-fat diet study, where they show higher expression of HIF1 alpha in response to short-term high-fat diet study. And we questioned whether this uptake of our lipoproteins may somehow alter HIF1 expression. First, we checked in the tissue, where you have lower HIF1 alpha immunoactivity, which is quantified here, and also of the target protein, especially PDK1, was lower. So in vivo, it seems that there's an effect, and then we started to analyze this in more detail in vitro. And here we use a cell line which is able to proliferate into brown adipose tissue, brown adipocytes, and they were treated with triglyceride-rich lipoproteins in the absence or presence of an inhibitor for lysosomal acid lipase. And then we did immunostaining for HIF1. And here you can clearly see that under control conditions, we have low HIF-1 alpha, but triglyceride-rich lipoprotein supplementation induce the protein abundance of HIF-1 alpha, and this effect was totally blocked by this inhibitor for lysosomal acid lipase, and the same pattern you can see also for this target protein PDK1. Then we did the same experiment with an HIF inhibitor, where you easily see that this somehow is the same pattern. With triglycerides, you can see again the increase, which was blunted with the HIF-1 inhibitor. And of course, we then analyze how triglyceride-rich lipoprotein could be important to induce the abundance of HIF-1 alpha in endothelial cells. And this is a typical model: hypoxia, low, you have high HIF-1 alpha; normoxia, you have low HIF-1 alpha. But what has been shown is that also reactive oxygen species, which are produced during beta oxidation, could stabilize HIF-1 alpha protein. And this we then tested in our model with the triglyceride-rich lipoproteins, because of course this triglyceride-rich lipoproteins generates free fatty acids after activation by lipolysis via lysosomal acid lipase. And of course, these fatty acids, before they can be beta oxidized, they need to be transported into the mitochondria. And this is done by the carnitine palmitoyl transferase 1. And this process, this can be blocked with etomoxia. And this we did. And here you can see the results. So treatment with TRL stabilized again HIF-1 alpha, and this was totally blocked with etomoxia treatment. And of course, to show that really reactive oxygen species are determining this effect, we also use another inhibitor, which is known as mitoQ, and this inhibits the production of ROS specifically in the mitochondria. And again, the same setup, you can see this effect is blocked. So I see I'm a little bit late, so maybe I will skip this slide, but maybe I can leave you with this model, which it's was generated based on this data, where you can see that in the capillary lumen, the triglyceride-rich lipoproteins are processed by lipoprotein lipase. Then, of course, the free fatty acids can be transported, which is not shown here on this model. But here we focus on the uptake of entire lipoproteins, which we showed is depending on CD36. And that these entire lipoprotein particles, which are internalized and transported via the endosomal route, are then hydrolyzed by the lysosomal acid lipase. Free fatty acids are released, oxidized in the beta oxidation, which generate ROS, and these ROS stabilize HIF-1 alpha, which is important for the thermogenic adaptation in acute cold by BAT remodeling and energy uptake, and under chronic cold for Watt Browning processes. And last but not least, here are the take-home messages from these studies. So the activation of brown adipose tissue really triggers the systemic changes in lipid lipoprotein metabolism. And this lowers plasma levels of triglycerides, remnant lipoproteins, and also atherosclerosis. We demonstrated a novel axis between white adipose tissue, beta cells, and brown adipose tissue, which is essential for the energy replenishment and thermogenesis in brown adipose tissue. And last but not least, we also show that lipoprotein particle uptake into endothelial cells of brown adipose tissue is essential for adaptation of thermogenic adipose tissues to sustained cold exposure. And with this, I'm at the end. And finally, I just want to show you Hamburg, because we have a very nice new building. This is the Elbphilharmonie. And if you are visiting once Hamburg, then please go to this building. It's wonderful. And maybe you give me also a note so that I can invite you for a seminar at our hospital. And of course, I have also to acknowledge all the people doing this work. And you already named Alexander Bartelt. So he's named now Professor in Munich, but he was a PhD student and postdoc also in my lab. And as the data I presented today, they were primarily performed by Alexander Fischer, he was responsible on the LRL story. He's meanwhile in Boston in Bob Ferry's lab and Markus Heine, and he's still in my lab, fortunately, so he can still do some cool stuff. And of course, we have a lot of different collaborations. So I will not go into the detail now because I'm already a little bit late. So I will stop here, and I'm looking forward to take your questions.
Thank you so much, Jörg. This was like a tour of the field, so it was amazing. We're going to take some questions, and we just raise hands, and I see Adil's hand as the first one.
Thanks. That was a really, really amazing talk. I have two questions. The first is probably a naive question. I was just thinking about some of the data that you presented in your introduction. And so are there humans with absolutely no BAT? I think you presented, that was like I think it was a letter from Nature Medicine. I'm just wondering if there are humans with absolutely like no BAT content whatsoever.
So this is, of course, a very essential and very important question. And actually, we don't know. Because, of course, brown adipose tissue is always calculated based on the activity with regard to glucose uptake via PET-CT studies. And of course, even though we know that, for instance, type 2 diabetic patients, they have very low activity for brown adipose tissue, we do not really know whether it's there or not. So it's maybe just insulin resistance and cannot internalize, cannot be activated, and just cannot internalize, cannot be found by PET-CT studies. So this is not known, and it's also not known whether it can be reactivated to sustain, maybe to improve metabolic health after some caloric restrictions or sustained cold exposures or something like this.
And quickly, my second question. So I'm thinking more in terms of some of your initial summary slides where you show the effect of insulin on the BAT. I'm wondering if the insulin also has an effect on, sort of inducing a transient activated state in the endothelial cells, kind of signaling them to uptake the lipids and then process them and then give them to the BAT.
Actually, we did in this study, we compared mice which lack the insulin receptor in the adipocytes with mice which lack insulin receptor in endothelial cells. And these data on the endothelial cells were not in the paper, but we didn't see any effect on glucose and lipid uptake in the mice lacking insulin receptor in endothelial cells. So actually, this, of course, this would be super cool, but we didn't see it. So, but in contrast, the insulin receptor signaling in the adipose tissue was essential.
Thank you, Adil. And Han-Rui?
Hi, this is Han-Rui from Columbia University. The results are really intriguing. Thank you. Part of my lab studies the role of macrophage lipid in atherosclerosis. So after listening to your talk and of course reading your paper, I just wonder if you have any thoughts on what the phenotype would be if lipase in endothelial cells are to be overexpressed, or in other words, do you think the system has the dynamic range that further boosting the lipase activity would further enhance the entire signaling cascade, as you're seeing in the knockout?
Good question. I don't know. So usually the lysosomal, the lysosomal enzymes, they are not really critical with regard to their activity, because they have so high activity that they usually are able to process everything which are transported to the lysosomes. Only if you really have a activity which is lower than 10%, then you can see something also in humans. So it's not really known whether overexpression would lead to a higher rate of activity. But if you may, what I would think, if the composition of the lipoproteins probably will influence also the activity with regard to ROS production. And of course, ROS production is one pathway, but there are a number of different pathways which probably are influenced by lipid uptake into endothelial cells. For instance, you could imagine, and I think, for instance, I see mad lines here, is the production of lipokines, which could be depending on lipoprotein particle uptake into the brown adipose tissue, or also on the in endothelial cells, of course, of the heart tissue, because you know, under these conditions of cold exposure, you have also massive changes in the lipid and energy uptake by the heart. So there's always an inverse substrate steal, I would say it in that words, between heart endothelial cells and endothelial cells of the brown adipose tissue. And this connection is not really good, or it's understudied. And also, if you are studying macrophages, I have to say this is also super interesting, because we do see also under cold exposure very high uptake into macrophages, as you have seen maybe on my data slide. So this is also not really known whether this influences the activity of the macrophages, which are also important to maintain the metabolic health of brown adipose tissue.
Great, thank you for sharing your insights. Yeah, I guess partly it's partly related to the local substrate availability as well, right?
Yeah, sure.
Thank you.
Thank you, Hanrui. Mabruka?
Yeah, that's really very beautiful talk. I enjoyed the data and the science. So I want to know how much it's to be called before you see the phenotype, and also is it sustainable? In people move between cold and from extremely cold conditions to extremely heat conditions. Is there any the effect is sustainable or not? And last questions, does HIF1 alpha regulate UCB1? And this is how you see the changes with HIF1 alpha.
So for the first question, so we very carefully actually use different time regimens to study how fast is the effect of cold exposure on the lipid uptake. And you really see it already after 30 minutes. So if you expose a mouse to cold, then it's immediately activated. If it's pre-activated, this is important, because if you house a mouse which at thermoneutrality and then expose it to the cold, then it will die because it cannot adapt to the cold. But if it's at normal ambient room temperature, which is 22 or 24 degrees, expose them to the cold, then you see after 30 minutes already activation. So we didn't study it earlier, so probably it's already after 5 minutes or 10 minutes, but you also turn it off quite fast. So if you then go back to the warm, it's also turned off immediately. So that is why we have to do some of our studies, at least if we are doing in a very time restricted manner, we have to do also the turnover studies in the cold room, which is not really nice for the people doing this kind of work. But in principle, if you have a sustained cold exposure, then you have also the activity is somewhat longer with regard to replenishment of energy substrates into the brown adipose tissue. But again, even if you have a prolonged cold exposure and then put mice back to for 2 hours or 4 hours to warm conditions, then you do not have the effect on the uptake anymore. And also heat production is lower.
That's interesting. And what about UCB1? I mean, there's any kind sort of...
So actually what we do see, so this is a awesome question. I cannot give you really a full answer because we didn't study it in detail. So it's not a direct target. What we believe is that there is a kind of endothelial differentiation towards brown adipocytes. This has been shown by a number of studies before. And what I do think is that due to the induction of this proliferation and angiogenesis process, that you also induced differentiation of endothelial or precursor cells which are in the endothelial niche, and these then get to the induction of UCP1. So I think it's a stepwise process, and it's rather the second step that you get an induction of UCP1.
Thank you so much.
Thank you, Margaret. Claudia?
Great, thank you. Jörg, that was a really fantastic talk. I really enjoyed that.
Hi, Claudio.
So my
Nice
question
to
is
see you
related.
again. Last time we saw on my last
that-
meeting, we met. That was the last meeting before Corona, the Keystone meeting.
Yeah, I think that was one of my last ones too, unfortunately.
Yeah.
Hopefully we can get back to traveling again and see each other.
Yeah, that's
but yeah, that was really beautiful. I really enjoyed that. So so you showed that the chylomicrons end up in lysosomal, in lysosomes, right? And so how do you make chylomicron remnants if that's the case? How do they get out?
How do they get out of the lysosomes? Or
Yeah.
This question. So if the chylomicron remnants are within the lysosomal compartment, then they got hydrolyzed by the lysosomal acid lipase. So then the triglycerides as well as the cholesterol esters are hydrolyzed.
Now press through the big toe mound to the right with straight movement. This time up to front. Good. Now reach your
So-
back to your toes.
Sorry. So can you hear me again?
We hear you, but we hear someone else too.
That's not me.
Yeah. Okay, well, it's gone now.
Late here, nobody is in the lab anymore. So
Yeah. Yeah.
So
it's gone.
so the free fatty acids are then used for better oxidation by the usual pathways. And of course, there are also proteins that are hydrolyzed by proteases, which also lead to the release of amino acids. But there's a lot of space, of course, to analyze this processing of lipoprotein particles by endothelial cells in more detail, also with regard to other components of the lipoprotein particles, which, for instance, could be used for lipokine production or for something else.
So do you imagine that once it goes into the lysosome, the chylomicron is disassembled and then it's put back together and then sent out into circulation to make
No.
to have chylomicron remnants?
No. So, of course, part of the chylomicrons which have been generated in the vascular lumen, they are then released and they are internalized by the liver. But the other part, which are internalized by the endothelial cells and transported to the lysosomes, they are hydrolyzed by lipases, lysosomal acid lipases, but also by proteases. And then you have this degradation products such as fatty acids, but also cholesterol and amino acids, which are then used probably for the proliferation process during angiogenesis.
Okay,
Does
got
answer
it.
your question?
Thank you.
Yeah. Thanks, Claudia. Matt is here. Yeah.
Hi, I'm Matt Lyons. I'm from the Joslin Diabetes Center. Thanks to the organizers for organizing this and let me crash your Zoom. Hey, Jörg, how's it going? Hey, I want to know, why do you think that other tissues don't take up glucose? Like, why don't other insulin-sensitive tissues take up glucose in the PET scans of those people? Why is it only the brown fat?
Of course, the activity of the brown fat is, of course, very high. This is one explanation. But the other thing is that I think what is often not really analyzed under these conditions, and this we get aware when we study this cold exposure experiments with our glucose tracer, because if it's activated by insulin receptor signals, then of course you would expect that it's also internalized into the muscle, for instance. And this we do not see. And this I think we can explain via the blood flow, because you do not really, you don't only need the the uptake by the cells, but you need also increased blood flow into the tissues that you can, into the capillary system, that you can also get the internalization of your energy substrates. And you always need both. So that is why you need adrenergic signaling and insulin signaling at the same time, which is somehow difficult to understand because this is catabolic and anabolic at one time. And that is, but you need both signaling pathways to have one, the increased blood flow, and on the other hand, then the activation of these anabolic pathways by the cells to replenish the energy substrates, at least under short-term cold exposure experiments. If you have long-term cold exposure, then adrenergic signaling seems to also to activate the energy uptake processes together with increasing the blood flow. But I think the important answer to your question is why we do not see other uptake into other tissues is the blood flow, regulation on blood flow.
Okay. Thanks. Nice to see you, bud.
Yeah.
Thanks, Matt. Ada, and that'll be our last question.
Yes. So my question is about the endothelial cells, and I'm kind of wondering, one, if you see any uptake of those lipoproteins in other organs, in endothelial cells in other organs, maybe kind of relates to the last question. And I also wonder about the CD31 expression that you see that is kind of fading away with time. And whether, yeah, you showed the mechanism of proliferation, but I was wondering also if you see other mechanisms of maybe like transdifferentiation or something like that.
So with regard to your first question, so we, by immune histochemistry, we checked also the heart tissue and also muscle for uptake of lipoprotein particles into endothelial cells. And we do see under certain conditions much lower, but some uptake into endothelial cells into the heart. I never thought in the muscle, which may be a problem of sensitivity, but this I don't know, but it's not a lot. So I assume that most of the fatty acids, the energy substrates which are delivered to the muscle are really fatty acids released by the lipoprotein lipase, and there's not much of lipoprotein particle uptake. What I found very interesting, if you have sustained cold exposure and look by intravital microscopy in the white adipose tissue, where you can clearly differentiate by eye between white adipocytes and beige adipocytes, and you have next to it endothelial cells, you can see how endothelial cells, which are close to the beige adipocytes, they internalize entire lipoprotein particles, and in the same picture, endothelial cells, which are close to white adipocytes, they do not take it up. So this is super cool, I would say, but I have no clue how this works. But of course, this is, I think, quite intriguing to follow how this is really organized. And the second question was with regard to transdifferentiation. Of course, we do think that this entire process is important for differentiation of endothelial cell as well as precursors of brown adipocytes, which are also present in the endothelial cell niche. But this we didn't study so much so far. So this, of course, is something which we would like to continue to work on.
Thank you.
Well, that brings us to the end, and we're going to hand you over to the metabolism trainees, everyone.
Okay, thank you again.
Thank you, Jörg. All right, I'm making the trainees co-host.
Thank you.
Shall I stop this